Welcome everybody. This is the SCI Fine Chemicals Group and Young YCP's webinar series. This is the sixth webinar in the series. We're happy to have AstraZeneca sponsoring the webinar series. So it's my great pleasure to welcome you today. And we've got our first presentation. We'll give a few more details. So the webinar series being set up to facilitate the links between organic chemists in industry and academia and provide a forum for discussion and for sharing information relevant to all aspects of the pure and applied synthetic chemistry, including medicinal chemistry, agrochemicals, drug discovery, process, and many others. So today I shall be chairing from the FCG. I have my colleagues Fred Hancock and Caroline Lowe who will be doing the Q&A after the presentation. So in terms of the Q&A session, this will be following Douglas Kell's presentation. So during the presentation, attendees are in listen-only mode. If you want to ask a question, please tell us where you're from, and that'd be good to hear you. We've had several international attendees, and type your question within the questions box, which is on the right-hand side. During today's presentation, there'll also be the opportunity for a few polls. So we will share that with you later today, so you'll be able to have a question. Right. Just to once again say thanks again to our corporate partners, and today we're sponsored by AstraZeneca, so it's great to have them on board. Supporting this webinar series. So I can now introduce today's speaker, Professor Douglas Kell, who's going to be speaking on how drugs really get into cells, why passive bilayer diffusion is a myth. So Professor Douglas Kell CBE is currently at the University of Liverpool, where he's a research chair in systems biology. He'd previously been at UMIST at the University of Manchester, and he's worked on numerous areas, specifically coming up with the terminology of metabolome and about transporters and their role in getting drugs into cells. He also works in areas of synthetic biology, enzyme discovery, and he uses high throughput experimentation and AI to sort of help his research efforts. So if there's more details there that you can find on the link there, which is available on the internet, and you can also follow him on Twitter too. So just to give you the heads up on the next presentations coming up in the series. So we've got back-to-back talks from AZ, more process orientated, so they were to do with the route to AZD 5718. So we've got Matthew Burns and John Mark Henry. And then following on the 23rd of June, we have a medicinal chemistry talk looking at the discovery of fipronil, which is an MR antagonist for the treatment of cardiorenal diseases. And this is from Lars Barfacker from Bayer. So I hope you can join us for one of those webinars in the future. It's my great pleasure now to let Douglas come to the stage and give his presentation.
Thanks very much indeed, Jason. Thanks very much indeed for giving me the opportunity to speak to you today about really debunking what is something of a myth that nevertheless appears in all the textbooks, apropos how drugs get into cells through the portion of the membrane that they used to go into. So I'm going to start with some take-home messages. Firstly, actually, when you look at real membranes, there isn't much phospholipid membrane in real cells, and consequently the chances of things diffusing through it are actually quite small in the first place. Secondly, I'm going to suggest that the way to think about why things are taken up is determined not by physical chemistry, log P and the like, as is widely believed, but in fact by the biology which proteins are expressed. In particular, the fact that the family of transporters, known particularly as solute carriers or SLCs, are very heterogeneously expressed, and this alone is entirely sufficient to explain the very great heterogeneity of uptake that you see, including via the blood-brain barrier. And that in consequence, we need to look at drug uptake into cells as a systems biology problem. Now, of course, the reason we're interested in this kind of thing is to understand how to make efficacious and non-toxic drugs that work well to solve medical problems. And there is indeed, of course, a very vast literature on that. And there are essentially two main strategies for discovering a drug. Classically, people used a function-first approach, in which we would take a drug and do what amounted to a phenotypic screen, often at the level of an organism or certainly a tissue, find a drug that worked, and only then might we care if we understood the mechanism by which it worked. Of course, the very great advantage of that is if it works, you do have a working drug. By contrast, we moved in the more modern era to a mode of science in which we started with an idea, a hypothesis if you will, that such and such a gene or a target was important in the disease and that altering its activity would be a good idea. We could find something that did alter its activity, typically in vitro, and only then would we test in the organism. The consequence of this, of course, is that we have this pipeline of drug discovery in which we start with candidates which may run to the millions and end up with one or two that actually make it out the door. But starting from first into humans, i.e. phase 1 in pharma speak, we still lose an enormous number despite the fact that preclinical safety and clinical pharmacology and so forth have been done. And it seemed like it was going to be a good drug. This is referred to, as most of this audience will know perfectly well, as attrition. And it means that in practice, something of the order of 90%, actually 92% in the latest survey, of drugs that get to phase 1 do not, in fact, make it out of the door at ruinous cost, because the effective cost of developing a drug is tenfold higher than it otherwise would be when 90+% of them don't work. So that takes it from 250 million to 2.5 billion, which is a horrendous number. And of course, people have looked at the reasons for this attrition, and the general feeling is that gross pharmacokinetics and pharmacodynamics, and you'll see why later that I've underlined gross, are not seen as being so much of an issue nowadays as once they were, but that actually about 40% of the cause of attrition is lack of efficacy, and 40% or so is due to toxicity. And in my opinion, that follows from this move towards an in vitro start rather than an in vivo start. And as I say, this makes it a systems biology problem. And the way we attack systems biology problems at the level of metabolism, where I've got the standard wall chart as a backdrop, is to make an in silico version, a digital model of whatever you're interested in this case, metabolism. And we, some years ago now, 12 years ago, got together the community to do this for yeast, and we made a consensus in which all the people who were working on yeast metabolic networks that we could get together agreed on what it was, and we made it available to everyone. And then a few years later, we did the same, or mostly the same crowd did the same for the human metabolic network, noting in particular that a third of the reactions in the human metabolic network, the things concerned with small molecule transactions, are transporters. And that's really quite a huge fraction of the whole. And this network seemed to work very well because it could predict a variety of things that turn out to be true. So now we're going to spend a bit of time about how things cross membranes in general, and those things in particular being pharmaceutical drugs. And our first foray into this field was, again, about a dozen years ago, where we started looking at the main two means by which drugs could get into cells by crossing a membrane. I'm not talking at all about endocytosis or anything like that here. And the two main mechanisms are diffusion through the bilayer, if such exists, and uptake through proteins that happen to be there, and those proteins are known as transporters. And what we concluded from that was, at the time, was that although everyone seemed to believe that stuff floated through the bilayer, we kept finding literally hundreds of papers in which they said, but as well as this bilayer transport, which by the way we never measure, we also found that such and such a transporter was involved in such and such a drug uptake. And when we passed 300 papers that said this, we decided that this was actually really not an exception, but actually probably the rule. And now we come to this dreadful word passive that is usually attached to the word diffusion and sometimes attached to the word permeability. And it is grotesquely misused, so much so that we really ask people not to use it at all to solve the problem. It has two meanings, or two meanings in common usage. The first is a thermodynamic statement. It says that the free concentration of drug inside and outside at equilibrium is the same. It is an equilibrative transport activity rather than a concentrative one. Two perfectly good words to describe that concept, which we ask people to use. There is also a mechanistic usage, which of course thermodynamics has nothing to say about, usually implying through or across a bilayer. It doesn't always admit that intended meaning, it just assumes it. And in the worst cases, which are surprisingly and shockingly common, it's actually conflated with the thermodynamic usage. So equilibrative, which is a thermodynamic concept, is taken to mean through the bilayer. In particular, we dislike the concept of so-called passive permeability because that hides even the distinction and conflates them all into one, because it doesn't tell us which of those uses of the term it wishes us to believe is going on, and that makes it confusion even worse confounded when you couple the word passive to the word permeability. And since we've got perfectly good words for each of these concepts, I encourage people to use them. If something is transporter mediated and concentrative, we call it active transport because that's what it's called. If it is merely equilibrative but transported, we call it facilitated diffusion and have done so for over 60 years, actually about 80. Whereas if you want your listeners to believe that the transport is lipoidal, call it lipoidal, call it bilayer diffusion. If you want us to believe it's concentrative, call it concentrative bilayer diffusion. If you want us to believe it's equilibrative, call it that. So, having dealt with the terminology, and I'm going to try not to use that word passive ever again, why is it that we think, and the textbooks would have us believe, that most of this drug transport goes through what bilayer portions of the membrane exist? And much of the problem hinges on this iconic picture from the famous fluid mosaic model membrane of Singer and Nicolson 72. That picture is taken from it in Science, in which by inspection the protein to lipid ratio is 1 protein to 10 lipids roughly. Now it's not actually hard to do a measurement of the protein and lipid composition of a membrane. Here's a prettier version of that same picture showing about 1 to 10. The real ones, when you do the actual measurement, are something like 3 proteins to 1 lipid by mass. The proteins stick out, roughly 1 to 1 by area. And it's not a hard measurement to do. What it means, though, is that there's next to no bilayer in real membranes, which is the kind we're interested in. Nothing I say today has anything really to say about measurements in black lipid membranes, liposomes, pamper, or any of the other kinds of artificial things that people have developed to study transport that are not real membranes. I only have to say about real biological membranes in real cells because that's where we want our drugs to act. Looking at that membrane, we immediately recognize that given that there are so many proteins in a membrane, and that could be mitochondrial or plasma, it doesn't matter for these purposes, the human genome is known to encode of the order of 1,000 different transporters of various kinds. In humans, these include ion transporters and nerve transmitters and stuff like that we're slightly less interested in. The kind we're particularly interested in is about half of those, the solute carriers, of which there are approximately 500. And these are influxes and antiporters, and there are also about 50 ATP binding cassette transporters, ABCs, who tend to be effluxes. It's about 4% of the genes in the human genome are involved in transport, 1,000 out of 25,000, and you can see them all at bioparadigms.org. So if you look at a real membrane, well, this is a cartoon, but drawn to scale, where the protein to lipid ratio is now 3:1, it's fairly obvious that there really is next to no lipid bilayer that is not influenced by the presence of a nearby protein. And when you look at the size of a classical small molecule, Lipitor, the biggest selling small molecule of its day, and probably among the biggest ones now, you can see that the chances of that fighting its way through a hydrophilic phospholipid head group, then through a hydrophobic interior, and then back through a hydrophilic head group are precisely negligible. And in fact, we know, of course, that this drug goes through a transporter mainly expressed in the liver, called in the old speak OATP1B1, organic anion transport protein, in modern terminology SLC solute carrier O1B1. All the others have numbers, this one is called O. And if we look now using molecular dynamics, which is ab initio way of determining by calculation what's going on, here's a picture of a real membrane containing aquaporin. The phospholipid head groups are yellow, the phospholipid tails are green, the aquaporin is those blue helices, and the red and white balls are water molecules. And it's immediately clear that there aren't any water molecules going through the green-tailed phospholipid bilayers. And I asked my friend who works on this stuff why not, and he said, well, don't be silly, that would be like asking them to go through butter. There is no permeability of even water molecules through the bilayer as calculated using molecular dynamics. In the literature, you will find two broad views. One has moved from a slightly more extreme version in which the transporters really hardly had any role, to now saying that they're equally important. That's the top view. And our view, which is, and I will give you the evidence for it, that the actual transport of drugs via phospholipid bilayers in any real membranes is negligible, and we call that PBIN for PBIN. And it's an oddity why people think that it might be real, and it's a logical anomaly, and I illustrate it first as follows, by analogy with the understanding of how to get good corn yields, or maize as we call it in the UK, in Aztec, Inca and Mayan Mesoamerican agriculture. So this was hypothesis driven. You'll see that I don't like these kinds of things by and large. And the hypothesis was that the maize god had to be placated to get a good yield of corn. And in order to do this, you had to make a sacrifice of a small fish. Actually, anchovies were surprisingly prevalent there, at least from the coast. And if you sacrificed a small fish at the time you planted this, the maize kernel, you would indeed get a good yield. And if you didn't, you would get a poor yield. And fairly clearly, you can guess correctly that that's exactly how it turned out. And people then inferred that the data were indeed consistent with the hypothesis. And that's the hypothesis, is that the maize god is important for giving you a good yield. Even though the maize god was never measured, all you measured was the input and the output, which fairly obviously you can see that there are other interpretations possible, one fairly obvious one being that maize is a nitrogen-hungry plant and requires a source of nitrogen to grow well. The logical structure involved in the corn god is exactly the same as the logical structure in believing that stuff gets into cells through the bilayer. Here the hypothesis is that bilayer diffusion is important rather than the maize god, and we do a measurement at the beginning, which is drug added outside, We do a measurement at the end, which is drug inside the cells, and we make an inference without ever measuring the mechanism that diffusion through the bilayer is important for getting that drug from outside to inside. And that structure, which obviously is false at best in the case of the corn god, is exactly the same structure. It's called in Latin post hoc ergo propter hoc, and it's obviously equally wrong. You cannot infer mechanisms simply by measuring outcomes. And our position since 2008 has hardened so that we now say, because of this, there isn't actually any evidence at all when you define evidence as data plus a sane theory and interpretation, that there is any significant drug permeability through undamaged biomembranes. And actually, it's very hard to design an experiment that might tell you it. You might think that varying the nature of the phospholipids would do that, or the amounts, but actually, because we've seen that the phospholipids interact with the protein, the phospholipids also affect the rate of protein-mediated transport. So that experiment can't tell you either. And as I usually say, I hope you'll begin to see as the talk progresses that you've probably had some data that aren't easily explained on the idea that stuff floats willy-nilly in and out of cells through the bilayer, but could then be interpretable from the point of view of transporter-mediated uptake. And if you do want to believe in lipid-only theories, you've got to explain to me why drugs don't get across the blood-brain barrier or a variety of other cells, despite the fact that the lipids are for practical purposes the same. And as well as blood-brain barrier, I could have said tissue distributions and species distributions more widely. So now we're going to have the first of our poll questions. I'm going to read it out, and then the organisers are going to give you an opportunity to vote on which you think is the true answer to the question, do you think CO2 crosses intact cellular membranes by using a transporter? And there's 4 options, of which they're all mutually exclusive, really. No, it all just goes through the bilayer. You don't need a transporter. Why would you? It's such a small little apolar thing. 2, B, it probably goes to the bilayer. C, it probably needs a transporter. And D, yes, definitely it has to have a transporter because life's like that. So I'm going to leave the poll organizer to let you vote accordingly and tell me when I can tell you what the answer is.
Okay, I'm going to close the poll now. So the majority went for answer C, 47%, 33% for D, 16% for B, and 5% for A.
Okay, well, you'll be pleased to know, of course it needs a transporter. I wouldn't be doing this talk if it didn't. And the transporter in this case is actually aquaporin, which, by the way, although not widely recognized, is also the transporter for oxygen. Oxygen doesn't cross either. And it's also the transporter, or various aquaporins are the transporters for nitric oxide. So even CO2 needs to have a transporter. 4 gigayears of evolution have ensured that everything needs a transporter. Now, one thing I'm often asked about is, how does your stuff relate to Chris Lipinski's rule of 5? I guess this audience will mostly know about the Lipinski rule of 5, which is actually 4 rules which happen to have numbers in them that are all multipliers of 5, which is why it's called the rule of 5. And many years ago, Chris Lipinski and his colleagues then at Pfizer recognized that people were wont to make terrible things that were either very hydrophilic and didn't go up, or were very hydrophobic and weren't soluble, and said that don't do it because they won't be absorbed and will have terrible pharmacokinetics, and that's the rule of 5. And I'm often asked, what does this have to say about my view of the world as to how drugs get into cells? And actually, Chris Lipinski never said anything about the mechanism of uptake that his rules might have implied. He did say that his rules didn't count if transporters were involved, which implicitly might suggest that he assumes that most of the time they're not. But nowhere did I find that the Lipinski rules were ever interpreted by their originators as saying that that therefore means stuff must float through the bilayer. And I put a picture of an Abbott drug in development here called navitoclax, which has a molecular weight of over 1,000 and 7 ring systems. And how that is supposed to pass through a bilayer is all I say is good luck with that. But the question is, why do people think that small non-electrolytes do cross through bilayers? And this goes all the way back to Overton in 1899, who measured the permeability coefficients of non-electrolytes across a variety of biomembranes and showed that in those days they used the olive oil water partition coefficient. Nowadays we use octanol. Showed that there was a very good correlation. I'm not going to show Overton's data. I'm going to show something from a little more recent, which has the same result, that when you plot the rate of uptake of non-electrolytes across biomembranes, you get a very good correlation over 5 or 6 orders of magnitude with the hydrophobicity as judged by, in our case, octanol water partition coefficient, which people then interpret as being consistent with the fact that stuff crosses through the bilayers. Now, actually, in many other cases, when you do similar things with drugs, you don't see any kind of relationship with real membranes, and the rate of permeation of, in this case, these drugs through KCO2 cells and the log K that is there. I haven't bothered to try and draw a line of best fit across that. It's actually very hard to throw random dots at a bit of paper and get an R squared as low as these people managed to get. There's no relationship between KCO2 uptake and permeability when you don't pick and choose your drugs. And the point is that these kinds of theories, log P-based theories, if you will, are biophysical, lipid-only theories. And there's another area that also had exactly such a theory where we now know that it was wrong. And I'm going to tell you briefly about that. And that's the mode of action and potency of general anesthetics, which pharmacologically are properly called narcotics. And the same Overton, actually in the same year, 1899, also found that the potency of a general anesthetic correlates very strongly, we'll see some data shortly, with log P again, to a cutoff, and the assumption was that they too, because there was such a negligible structure-activity relationship, it was assumed that they too acted in some sort of biophysical way. Of course, in those days we didn't know about bilayer membranes, and we certainly didn't know about nerve channels. But in the more modern interpretation that we'll look at on the next slide, it was assumed that by partitioning into membranes, they would squeeze the nerve channels. So here is a plot of anesthetic potency against the ability of a bunch of molecules to partition into a membrane. And again, over 5 or 6 orders of magnitude, there's a very good correlation. The more hydrophobic ones are much more potent. And now on the next slide, what I'm going to show you is the same molecules, but in a different assay. And actually the correlation is even better, but the assay is the ability to inhibit a soluble enzyme. No membrane is present at all. That enzyme is luciferase, whose activity is easily measured by light emission. And you can see, if anything, there's an even better correlation between the potency of various small molecules, including anesthetics such as halothane, and the ability to inhibit this enzyme. And because it's a small soluble enzyme, its structure's been done, and the binding pocket for these kinds of molecules, in this case bromoform, is known. And so all it's telling us is that there's actually a hydrophobic binding pocket in the protein that, from a biophysical point of view, which remember can't tell us mechanisms, looks rather like partitioning into a lipid of some kind. And from a biophysical point of view, the forces that you need to enter and exit from water to a low dielectric, such as a membrane, are no different from those to do the same into the hydrophobic pocket of a protein, and they involve making and breaking hydrogen bonds, so-called hydrophobic interactions, and also there would be polarization and polarizability terms for those who follow the Abraham theory of solvency. So biophysics alone cannot tell us about mechanisms because it's essentially thermodynamic kinds of statements. What can tell us about mechanisms is varying the biology. So here's an experiment in which we look at the anesthetic potency of halothane in a real mouse standing on a thing that it can fall off as it gets anesthetized, and the wild type at about 1% halothane is completely incapable of moving, whereas a knockout in a particular potassium channel called TREC1 at the same amount of halothane is perfectly fine and doesn't even notice it's there. The simple and indeed accurate interpretation being that anesthesia is dependent, at least in part, on the presence of this potassium channel, and without that channel, anesthesia happens far less readily. And I can tell you that if you'd used a different kind of anesthetic, an injection anesthetic, you would have found GABA A receptors are involved, and if you'd used a very recondite one, which is the gas xenon, you might have found that glycine receptors were involved. In other words, they're all proteins. We don't need lipids to have anything to do with it. And now, as I've mentioned before, if lipids were the story, you wouldn't expect to see any differences between different cells because for practical purposes, the lipids are very similar. But as we will see, there's going to be some very big differences. There's lots of stuff on this slide. I'm not going to read through it all. I'm going to concentrate on two of the things that are on this slide. First of all, the recognition that actually even so-called lipophilic cations that were thought to flow happily through phospholipid bilayers require transporters. And then we will look briefly at the fact that drugs do concentrate in specific tissues, which they wouldn't easily be expected to do if they just floated in and out. So here's a molecule. It's referred to as a lipophilic cation, dibenzyl dimethyl ammonium. There's an N in the middle of that, hard to read, I'm afraid. And I've not showing you what its transport system is. And I think you could look at that molecule if you don't know the answer for a long time, and you wouldn't guess which transporter that is there, and it indeed has to be there, it is using, and I will tell you the answer is, it's using in yeast the thiamine transporter. Thiamine normally thought of as a water-soluble hydrophilic vitamin, unlike, say, vitamin D or E, which are lipid-soluble, and thiamine is taken up into yeast cells via thiamine transporter and does not pass through a lipid bilayer, and neither does dibenzyl dimethyl ammonium, because if you get rid of the transporter, it doesn't do it. And you can also find an inhibitor, which is called dithiamine. Now, quite recently, I'll just show you some of our own recent experiments. We've been looking at the uptake of cations into the bacterium Escherichia coli, and we've made our life easy because we're going to use fluorescent dyes whose uptake is easily measured. And I'm going to show you on the next slide two of them that are there. The first is usually referred to as carbocyanin. It's got a long name which is written there, and it is a cation, and it is considered to be taken up in response to membrane energization. Those who believe in chemiosmotic coupling will say in response to a membrane potential. I'm not among them. And cybergreen, which is also a lipophilic cation, and stains, and indeed its fluorescence is very greatly enhanced when it stains double-stranded DNA, is another. And they both contain, interestingly, this benzothiazole moiety. Carbocyanin has two of them. And we're going to measure the uptake by flow cytometry, which I guess most of you know what that is, but if not, I'll just briefly rehearse. you have a sample which is injected into a faster moving sheath fluid, and it's interrogated by a laser, and if there are fluorescent molecules, they fluoresce, and if the cell comes through, it scatters light, and that triggers the detection of any fluorescence as well. And there's a huge palette of possible fluorophores that we use. And if you look now in E. coli, the nice thing is that you can get knockout versions of almost every gene and all of the non-essential genes. With no dye, we get a fluorescence that peaks at about 5 × 10⁻³ If we take a knockout of an efflux protein, it's about 100-fold more, something more like 5 × 10⁵. The wild type, and we've almost set the amount of dye added to give approximately an order of magnitude more than the no dye, is around about 5 × 10⁴. And here's one with a knockout of the ATP synthase subunit. So you can see this quite huge variation of uptake of the same dye into different cells that differ only in the presence of a single gene. And there's about 4,000 genes in E. coli, and in this case we've picked about 500, which are transporters, and various other ones we're kind of interested in. The wild type, this averages uptake is around here, and here these tend to be effluxes, and here these tend to be influxes, and you can see that there's an enormous variation, a 36-fold variation, which means that the lowest to the highest is 36-fold. That means that less than 3% of the uptake could possibly be going through carriers at all. Of course, it's going to be even less than that. If you're pink in this picture, you're a so-called Y gene, which means you're a gene of unknown function, and that's telling us how little we know still about the genes of unknown function. Then we're going to do the same with cyber green, but to make it quicker, I'm just going to plot for each individual knockout strain the mode uptake of the cyber green versus the carbocyanin. And you can see again, despite having quite similar elements of the same structure, the benzothiazole, there is negligible relationship between the uptake of those two things. Huge number of these Y genes for the diac3 in particular, and no particularly obvious reason a priori why these things are so different, but this massive variation, 70-fold in the case of the cyber green, massive variation in single gene knockouts of E. coli. It's telling you, and the lipids are practically the same every time, that lipids have got nothing to do with it. Just give you one mammalian example from a drug example. It's a drug that didn't actually make it out through the door, called casopitant or Zunriza, and you can see the variation in uptake between blood and liver, say, is more than 50-fold. And I don't know why it was withdrawn, but it's a reasonable bet that when you're 50 times more concentrated in the liver than in the blood, you just might have a chance of being toxic. At all events, these are very large variations in the uptake into individual tissues. Here's another one of our own recent experiments, unpublished, with Marina Wright-Muelas, where we look at the uptake of a whole bunch of things that we just use serum as the source of them, and we look to see what disappears from or appears in serum after, in this case, an hour, with 3 different cell lines. And you can see in the case of HEC, they excrete a lot and don't take up very many. Uptake is in blue, excretion in red. K562 is just a cancer cell line, more like 50-50, and Pank cells, another cancer cell line, take up a massive amount of things, whereas they excrete relatively little. Everything else about that experiment is the same, massively different amounts of uptake and efflux. It's all because of transporters. Here's another nice example from imaging mass spectrometry. On the bottom left, labeled C, is an ordinary micrograph of a mouse's kidney. That mouse has been dosed, as it says in the title, with the anti-cancer kinase inhibitor Imatinib, and we can then ask the question, using imaging mass spectrometry, what does that kidney look like, and you can false color it using lipid signatures. And when you do that, you then see 4 tissue subtypes in the kidney that any nephrologist would tell you in the textbooks are well-established tissue subtypes of a kidney. Because we've dosed the mouse with Imatinib, there's a particular molecular ion that we can detect, or a particular mass ion that we can detect from the Imatinib, shown in green at 494.27 mass units, and we're going to now see where that was in the kidney, and the answer is in only 1 of those 4 tissue subtypes does the Imatinib get taken up. No theory that assumes that most of the uptake is due to lipids can possibly explain these kinds of things. What does vary between tissues, however, is the expression of different proteins. You can go to the Human Protein Atlas, as I've done here, take any transporter you like, I just took the first one beginning with A, and look, in this case it's antibody staining, at the expression profile of, in this case, what is SLC7A3 in different tissues. And here the esophagus, loads of it, liver, none at all, and in these other 2 tissues, which are small intestine and duodenum, quite heterogeneous expression in different parts of the tissues. What is varying massively is not the lipids, but the protein expression levels, including transporters. Actually, they vary more than any other kinds of proteins amongst different cell types. So here's 2 livers, and if you remember only one thing about this talk, maybe let it be this slide. Both livers, as drawn, are intended to have the same total amount of uptake, but the liver on the left has a homogeneous amount of uptake. Its gross PK/PD, pharmacokinetics and pharmacodynamics, are simply measured by what goes in and what comes out. This liver is designed on the right to have the same gross pharmacokinetics and pharmacodynamics, but in this case, 2/3 of the cells don't take up any at all, and 1/3 takes up 3 times as much. Fairly obviously, the ones that don't take any up, that drug's not going to have an awfully large amount of efficacy because there isn't any there. And the one that's taken up 3 times as much, or I could have said 50 times as much, are going to have potential problems with toxicity. And no conventional PK/PD done at the level of an organ is going to tell you about that. You can only find that out when you start looking cell by cell, or even within cell by within cell in the worst cases. But the main point is very clear. Heterogeneity of uptake, as caused by heterogeneity of transporter distribution, will have a massive effect on the efficacy and toxicity of pharmaceutical drugs, and those, remember, were the 2 main causes of attrition, accounting for 80% of it. There are 2 other, there are many other, but 2 I'm going to concentrate on, other ramifications of the view that if drug transport is largely mediated. The first is that the transporters should be there for something, and maybe the something is moving endogenous metabolites around, and that the drugs might just look like metabolites. And we'll come to that second, but first we want to answer the question, how do we find out, if you believe any of this, which transporters are actually being used by particular drugs? And to attack that, and back in the day, we always used to do yeast, and this was a project I had with my long-term collaborator, Steve Oliver. The nice thing about yeast, like E. coli, is that you can get a variant, and it makes a haploid form, which just means 1 copy of the gene, of all the non-essential genes. And so the idea is very simple. If you can add enough drug that it kills the cell that lacks the transporter for that drug should survive, and you can therefore look to see by, in this case, we barcode the mutants, but nowadays you do it by RNA-seq, which cells survive. And this paper's 9 years old now. I'm not going to go through it in any more detail, just show you one example, which is in this case a drug called diphenyline iodonium, marked there, and in this case it tells us that the transporter that is most expressed in the ones that survive is one called NRT1, nicotinamide riboside transporter number 1. The delta means deletant, and so if we look at that one on its own rather than with all the others, here's the effect of that deletion on the growth rate of the yeast relative to the wild type, and in the presence of nicotinic acid, which was the only thing we could buy. We couldn't buy the riboside. Whereas the diphenyline iodonium in the black ones here completely clobbers the growth rate of the yeast, whereas the deletant binds far less, and the nicotinic acid is also protective, all entirely consistent with the view that if we wanted to know the transporter for diphenyline iodonium, it is the nicotinamide riboside transporter. So we've done our job. There's one caveat, which is if there are lots of transporters, say there's 10 transporters, all of which contribute, then knocking out one of them isn't really going to have much of an effect, and the others can take up the slack. So our method only works really well when there's a relatively limited number of transporters, which in fact is often the case. And here's a lovely example. In mammalian cells from my chum Giulio Superci-Furga, who was interested in this drug here, sepantronium bromide, also known as YM155. It's an anti-cancer drug in development. It's had 11 clinical trials, looks quite good. You can see that it might be a DNA intercalator. It also hits something called survivin. And he does exactly the same as we did. Indeed, acknowledges the idea having come from us, and asks the question in a haploid cell line of humans, which, when I add this drug, are the cell line that survives when the wild type does not. And he does it 122 times, and every time the cell line that survives is a deletion in this non-essential transporter called SLC35F2. And it's 200 to 500 times less sensitive than is the wild type, which is telling you that less than 0.5% of the drug can possibly go through anything except this transporter, including, obviously, the phospholipid bilayer. Really, for practical purposes, this is the overwhelmingly dominant transporter. And when you look to see the effect of knocking out something else after this, it's actually SLC35B2, and that knocks another tenfold off it. So you can then ask the question, in a whole bunch of different cell lines, how much do they express this transporter as judged by transcriptomics? And you can see there's something of the order of 10,000-fold variation on this log2 scale. And you can see that there's a strong variation in the potency of the drug in killing these cells as a function of the expression of the transporter. So the reason this drug looks quite good in some assays and terrible in others is simply the expression level of the transporter, and that's the beginning and the end of it. Now the good news is that the flip side is true. If you want to target a drug to a particular case, this is also a Pfizer paper, in fact, and in this case they're interested in inhibiting glucokinase, not glucokinase, selectively in the liver as an anti-diabetic drug, and in this case they can arrange that it's taken up by that same transporter that we saw before, SLCO1B1, only in the liver really, and massively accumulated there. So you can actually add quite small amounts, so it's not toxic anywhere else. And in a piece of our own work, we were asking the question, if there is a transporter for a drug of interest, in this case it's another anti-cancer drug called gemcitabine used in pancreatic cancer, can we add a second molecule that alone is not toxic, but when we add it manipulates the ability of the drug to be retained by the target cells and therefore gives you selective toxicity? And the answer again to that was yes, but it's all published, so I'll just draw your attention to the manuscript. I was also asked to speak briefly about the role of transporters in phenotypic assays. So as we looked at the beginning, a phenotypic assay is where you look at a cell or a tissue and the effects of the drug on the cell or tissue. And so it's fairly obvious that from the perspective of drug discovery, the good news is that transporters don't matter in phenotypic screen in the sense that they take it into account and they measure both transport and the effects of the drug you're interested in on all the targets simultaneously. In a sense, the bad news is the same. You don't know necessarily, now you may or may not care, but you don't know simply by adding a drug and doing a phenotypic screen if you're measuring its effect on the target mainly or on the transporter mainly. If it's very poorly transported, it might be fabulous on the target but just never get there. If it's very well transported or indeed concentrated, it might be terrible on the target but it may be concentrated up. And if you don't know that, you're going to be in trouble. If they only do one of these things, then you have the chance of getting into a bad state because your QSAR measurements will be measuring both the transport and the activity on whatever target you happen to think it is interacting with in vivo. And most things interact with at least 6 targets, by the way. So now we're going to have our second poll question, which is, what are the real substrates of these drug transporters? And I'm going to be quiet now while those questions are asked and you can see them on your poll screen.
Yeah, closing the poll now, and I'll share the results. You see that? So the endogenous metabolites, 85%, pharmaceutical drugs, 3%, endogenous natural products, 10%, and fluorescent compounds, 3%. So while I'll stop.
Great.
Hide that now.
Okay. Yeah. So our original assumption, we will look at this now. I'm going to tell you the real answer shortly. But our original assumption was that indeed, because we've seen all of those molecules, and this was published here, because we've seen all those molecules on the wall chart, it's reasonable that they have to have transporters, and it's reasonable that drugs might hitchhike on them. And we're going to call them, for these purposes, endogenites, for endogenous metabolites, to avoid the confusion of using the metabolite when people might think it means metabolite of the drug. When we use metabolite here, I mean endogenite, the endogenous metabolites that you would measure by metabolism. And as most people will know, in cheminformatics, we can ask the question, how similar are two drug molecules, by using various kinds of so-called fingerprints, in which we turn the drug structures into a string of ones and noughts and ask how many of those strings have a naught or a 1 in the same position. And obviously that can go as a fraction between 0 and 1. And when we do these two, we get a number, it's 0.485. That's using a particular encoding we'll meet in a second called max. Doesn't matter particularly what it does, it just counts up how many of the same functional groups are there. Here's another vitamin, riboflavin, and you get 2 other molecules numbers, and the main point is to show you that they don't all add up to 1 when you go around the circle. It's a pairwise comparison. You just do them one at a time against each other. Now we know all of the metabolites in the human metabolic network, not counting a lot of lipids that we weren't interested in. We call that Recon 2, and we know all the marketed drugs because you can get the list. And so we did the simple question, how similar do these marketed drugs look like to our endogenous metabolites? And it'll be easier to see on the next slide, but you can see that anything above 0.5 is in a sort of yellow to orange to red, and anything below is in various shades of blue. And you can kind of guess that mostly there's going to be at least one metabolite for which there is a drug that has a similarity above 0.5. And we're going to use the max encoding here. We'll come to all the others which are marked here. There's 8 different encodings on this slide. And we've rank ordered those drugs in order by the size or the greatest similarity of the drug to any metabolite, doesn't matter which one. As we go down our max encoding, by the time we get to 85% similarity, we've used up about 0.75 max encoding, and by the time we get to 95% of the drugs, 90%, all of the drugs are within a max similarity of 0.5. So as I said, these are numbers, but we'll come back to whether they make any sense or not. What we didn't ask at this time was, as we go down these different curves with the different encodings, is the rank order always the same, or does it vary somewhat? And the answer is, it varies like there's no tomorrow. Some drugs that are seen as the most similar in rank order in one encoding, one called layered in this case, are amongst the least similar as judged by Max that we saw, and vice versa. Here's one where Max thinks it's among the most similar, but rank order layered says it's amongst the least similar. And I'll just show you one more, and you can see that whatever pair of encodings I do, there's no major relationship other than at this top end, perhaps, where the encodings tend to give you something similar. And indeed, when you add up all the rank orders, you find there is this phenomenon in which, if you're very close to a metabolite of some kind, you tend to be reasonably close, whatever the encoding. But as you go further and further away, it tends to be just a mess, with a breakpoint somewhere around a Tanimoto similarity of 0.8 to 0.75. And any drug discovery person will tell you that if you have a molecule that's bioactive, there's a much higher chance, let's say 30-fold higher chance from random molecules, that it will also have bioactivity if it's within 0.8. So that's great. 0.8 is fairly similar, but unfortunately, that explains only 15% of my drugs being within 0.75 when I was being generous. Doesn't explain the other 85%. Now, there is a molecule, I haven't put it on the screen, so I'll just describe it, called ergothioneine. We're quite interested in it, actually. It's a nutraceutical, and it's found in mushrooms, and it happens that there is a transporter for it called SLC22A4 that humans have evolved specially to take it up. Now, ergothioneine is a natural product, so you begin to think, well, that's not unreasonable, actually. Maybe some of these things have evolved to take up natural products that are good for you. So you can do the same exercise where you compare drugs with the molecules in the metabolites in the natural products database. And when we do that, instead of being a miserable 15% as we saw before for a max nearest similarity of 0.75, we get up to 60%, which is much more respectable. And now remember that I said to you that actually, which was the best encoding was kind of, if not random, at least different for different molecules. There is no particular reason to choose any fixed encoding. Why not choose an encoding that is the best encoding when I try, in this case, 9 by now different encodings of the molecule and see if my natural product and my drug are similar to each other on any encoding. And under those circumstances, I can find a natural product that's close in Tanimoto similarity to any drug, 99.9 to 99.99% of them. The real substrates of most substrate solute transporters are in fact natural products, and that has quite some impact, in my opinion, on where you might go to try and discover new drugs. It certainly has an impact on our understanding of what the inverted commas real substrates of these drug transporters are, and why it is that they do take up drugs that they've never seen in their earthly lives that were dreamt up through the cleverness of the pharmaceutical industry. In fact, just as a heads up, I'm part of a large IMI consortium now from EU funding with Supertifurga, trying to look at the range of drugs, because many of these transporters, unsurprisingly, are orphans as well as the E. coli ones are. Conclusion. There isn't any doubt that there are many transporters and that we don't know their specificity, and in many cases they're almost certainly not for endogenous metabolites. I've tried to persuade you not only that drugs enter cells by hitchhiking on known transporters, but that actually they only do so. The consequence is that bilayer diffusion is both negligible and a myth. That means that we have to think of the uptake of drugs as a systems biology problem, as the PBK people have long done, I might say, and the consequence is that this has considerable implications for designing safe and efficacious drugs, and we need to understand pharmacology from a systems point of view. That means knowing which transporters are used, knowing what their specificities are, and knowing in which tissues they are expressed in a functional way. And just at the very end, I will put up this. You can find it again on my website or SIG file, because I ended up writing this book and putting it up. It's not quite finished, partly to stop Brexit, but partly it was driven by my failure to understand why people believed in things, including drug transport through bilayers, despite all the evidence against it. And it's actually, there's oddly enough some evolutionary reasons why human beings are susceptible, but I'll let you read the book and the references it cites for that. But with that, I thank you for your very kind attention.
Thank you very much, Douglas. That was a very thought-provoking presentation. We're going to move to a few questions. So we've got Fred and we've got Caroline online to help as the QA moderators. If I could just ask one quick question, Douglas. So it's very interesting, the role of the transporters potentially in PK/PD models. And I was wondering if you'd had any information in terms of tissue specificity. So say if you were looking at fibrosis and there's different types of fibrosis, where you've got any information on how drugs can be tissue specific because of the role of transporters.
Well, as far as we're concerned, they all are tissue selective, depending upon the role of transporters. There are relatively few transporters that are actually expressed in almost every tissue. Indeed, we published two papers, actually. The first is called Gene Gini, G-I-N-I. So Gini was an Italian economist who was interested in the distribution of income between individuals. And he came up with something non-parametric called the Gini coefficient. And you can use this exactly to describe the distribution of anything such as transporter expression between different tissues. So if your Gini coefficient is very large, it means you're really only expressed in one thing, whereas if it's very small, you're expressed homogeneously. And the Gini coefficients for transporters were amongst the largest for any class of compounds. It is a transporter that determines whether you're a tissue, a lung cell, or a kidney cell, or a liver cell, or what you will. So that's why we have to find out. In the case of SLCO1B1, the famous statin transporter, if you will, it's really only expressed in the liver. The next one down is like 2% of the liver expression levels. And so these things can account exactly, can and do account for all of that. It's only recently that people have realized this, though, that really this is actually not only the candidate explanation, but the explanation. You don't need to look further. Fibrosis can be caused by many things. That's another story. Yeah,
I think that's really interesting. I might pick up on that later, but we'll break to Caroline now. The next question for the audience.
You're muted, Caroline.
Probably helpful I unmuted myself, yeah.
Yep.
I have a couple of questions here from Sergio Chelis from Leeds. He started earlier on in your talk by saying, given the evidence that log P is not accurate enough to determine permeability, would you recommend drug discovery companies to think about changing their approach if they haven't yet? Which was followed by,
Well,
do you think there's enough research effort to determine drug carrier associations?
well, you won't be very surprised that my answer to the first one is yes. I mean, I think people will be expected to measure log P regardless because their managers will expect it. It's relatively unhelpful. I'd be moving towards lower log P because you might have some hope of specificity slightly more there. There tends to be more promiscuity with higher log P, but that's all. And do I think more research is needed? Well, obviously, I'm a research guy on transporters, so you won't be surprised that I think that this. But I'm trying to give evidence as to why I think it is worthwhile, not merely that I think that something I'm interested in is important.
So what's the best resource to go and look up these things? Can you find is there a way of predicting which compound will be transported by which transporter?
By and large, not. And indeed, that's part of what we hope will ultimately be the outcome of the IMI consortium. Bioparadigms.org lists all of the SLCs, and if it's known, and there's a whole review issue from 2013, which drugs are used, you can go to Drug Bank. Drug Bank's read some papers, so it's text mined them in a rather coarse way, and it can find associations. It doesn't fail to distinguish substrates from inhibitors. The bottom line is it's pretty virgin territory.
Fred?
Douglas, you have a question from Jorg Blankenstein at Sanofi. And he asks a question that came, I think that sort of follows on from your whole argument. Really, is how are biomolecules then treated by these transporters?
Well, they are substrates too. Not for all of them, of course, and as I've indicated, certainly in the case of, well, we don't know what the natural substrate is for the SLC35F2 that took up that YM155 drug. In the case of the SLC22A4, the only known substrates of any significance are ergothioneine and stachydrin, which is proline betaine, which is actually in citrus juices. There are a couple of drugs that are also taken up by that transporter as well. But by and large, people, the experiments haven't been done. There are known ones. So many of the, let's say, nitrogen-containing compounds, penicillins, cephalosporins, are taken up in the gut by what used to be called PEP-T1. It's SLC15 family in modern speak. Glucose transporters are well known, amino acid transporters, nucleoside transporters are SLC 28 and 29. One's equilibrative, one's concentrative. 28 is concentrative, I think, and 29 equilibrative. So the knowledge is becoming available, and there's to take the gemcitabine example, which is why we went and studied it in particular. There's a clear correlation between the efficacy of gemcitabine and the expression levels of the nucleoside transporters. Gemcitabine is a cytidine analog. It's not very surprising. Couldn't possibly get across without a transporter. Turns out the main thing that our thing did was to inhibit an efflux transporter, by the way, when we increased its toxicity. One in particular, not the transporter itself, but the expression level of the transporter. So there's lots of things out there that are going to be discoverable by varying the expression levels of transporters in a tissue selective way, just as the human body varies the expression level transporters in a tissue selective way.
I have a question here from Kathy from C4X Discovery, who says, how important do you think the shape and conformation of compounds is to recognition by transporters?
Well, since I used to be in Manchester, and in fact I had a Skype with one of C4X's founders this very morning in the shape of Andy Armand, I'm well aware of the importance of conformation in it, and it's something that's been very little studied with respect to SLCs, Kathy. So you know, the field is yours for people who have these techniques like C4X does, and it's really hardly been looked at, but bound to be no less important. I mean, a transporter is just another kind of a receptor. And actually understanding what makes a molecule an inhibitor, where it's bound but not transported, versus what makes a molecule a substrate, where it's bound and then is transported, is absolutely unclear in all except a very small number of cases. Really interesting area.
Douglas, we have a question from a delegate in the US, in Boston, Alex Hard. Does similarity to known endogenous metabolites, single or multiple methods, predict permeability of a random subset of small molecules?
I've slightly missed what the question was, Fred. I'm sorry.
Okay.
The similarity predicts for some things. So the short answer is everyone's almost bespoke. Some transporters are quite selective and will really only do one thing, like the SLC22A4, one or two things. Some are quite promiscuous. The PEP-T1s take up all kinds of things. Many of the effluxes, all kinds of things like PGP, of course, is well known. So the answer is knowing about one thing can be predictive, but what we need is a QSAR for every transporter. We're armed with a QSAR, you can then predict any molecule. That's what we need. I mean, it's as simple as that. I know it's a self-defining prophecy, but if you find out the QSARs for a transporter, then you're off to the races. All you need after that is where their expression profiles, and you can predict the tissue distributions.
So actually, if you want to target a particular organ in the body, would you suggest specifically looking at the transporters or the expression of particular transporters in that tissue?
Totally. And if you're interested in CNS drugs, they don't float through the bilayer either, but there are transporters in the brain. The brain would be dead if there wasn't. If you couldn't get glucose and amino acids in, and choline, and there's a few other known transporters. So you can add suitable groupings that will make things substrates for transporters and aim for some tissue targeting. Absolutely.
So another question from Jörg Blankenstein at Sanofi, who says, what is the relation of uptake with signaling molecules like hormones? Presumably they all have their own individual transporters.
Yeah, they do. They do. In some cases, and I can't immediately think of ones in my head, but of course these are quite hydrophobic, so the substrates tend to be sterols bound to proteins initially and then just released up the surface. So yep, bile acids, sterols, other hormones, yep, they've all got transporters. There's no floating through the bilayer of anything.
I have a question from Hannah Felsted from Southampton. How do you think carrier-mediated drug transport is affected by molecules that are able to change their conformation in an environment-dependent manner to facilitate intramolecular hydrogen bonding?
That's quite a generic question because, of course, it depends what the molecules are. But there's no a priori reason why there couldn't be some quite significant effects, because the transporter will bind certainly some particular subsets of conformations, and if you've done something to that small molecule that it doesn't adopt those conformations, then it will have less likelihood of going in. And of course, you could flip that and say if you might even find things that add more. I'm also asked in a similar vein, I don't know whether you had this question, how DMSO works, because sometimes adding DMSO improves the rate of uptake of things into cells. And everybody assumes, I think, that kind of implies that it knocks little holes in the membrane and loosens up the bilayer. No, it doesn't. What it does, the substrates of these molecules that aren't very soluble, that DMSO helps solubilize, is it's exactly that. they're forming absolutely microcrystals that are not substrates. And all this DMSO is doing is making the single molecules available to the transporters, which is what their substrates are.
As question here from Colin McKinnon, who says, I think a lot of people will be asking this, can you envisage an economically viable medium throughput screen for recombinant SLCs that could become part of a discovery chemistry department? Or does such a thing already exist?
So what I should have said when I put up the slide about the IMI consortium that's just had that sort of hello, we're there paper in Nature Chem Bio, is that part of that has made a knockout and overexpression variant in HEC293 cells of every single transporter and deposited them at AdGene, and you can get them now. So not only can I envisage it being as part of a medium or high throughput screen, I expect it to be taken up accordingly as a result of the availability of these things. Now, of course, the big companies have all got CRISPR-Cas abilities. They could knock these things up in afternoons, but you know, a relatively small consortium knocked up over 400 of these in one lab. So, you know, but by we I mean Giulio Superti-Furga's lab has made these available through AdGene. So absolutely, and of course, you know, we like these fluorescence assays, as does Pharma, which are high throughput, and in many cases, well, I didn't mention this other paper. You can find it on my website. And it's in bioRxiv. If you think about, many drugs actually contain aromatic rings, and part of the reason for that is that if you want to bind to something potently, if you're flopping about because you're a carbon single bond, binding causes a massive loss in entropy, which is giving you your delta G going the wrong way. Whereas if you're an aromatic thing, you can go splat and it doesn't care. So many drugs have aromatic rings, and so do many fluorophores, because the fluorescence is coming from the conjugation that you get from the aromatic rings. So actually, many drugs, in a Tanimoto similarity point of view, actually look like fluorophores and vice versa. We published a paper showing that. So there's nice fluorescent substrates capable for almost every drug.
Could you envisage having some sort of fragment system, you know, as in this particular fragment has a high affinity for this subset of receptors and this one for another subset of transporters?
Absolutely. It's a strangeness that fragment-based drug discovery has tended to concentrate on targets and receptors and NMR and modeling. Whereas if you look at the RO3 rules for fragments, one of which is a mass of under 300, actually, if you ask what fraction of marketed drugs have a mass of under 300, the answer is 25%. There's nothing particularly magic about being small. Of course, you can explore bigger parts of chemical space if you're bigger, but lots and lots of very small drugs that are absolutely fragment sized are well-known drugs. So I not only see it, but we've even published on that. So absolutely.
Right.
Douglas, we have a question from Kane Bastick at Durham University. Given your opinion on ligands and drugs binding to transporters, just as they bind to receptors, do you think the line between transporter and receptor is more blurred than we think? Should we be considering transporters as a subtype of receptor?
I certainly don't object to the notion. I mean, it's up to people whether they think it helpful or not to do so. Certainly they're both binding agents. I mean, I tend to think of them more as something that will take your drug inside to attack an intracellular target, whereas receptors, as we know, you don't have to go inside. You transmit your signal by other means. But I certainly wouldn't have any objection in principle to recognizing the similarity like that.
Okay, I think we've.
I had a question. Oh, sorry. I was just a quick one. Yes, sorry. I was just curious about you mentioned about entropy. What do you think? What is supplying the energy to this transport? I mean, you know, maybe pick a few typical examples. What is the energy that's being supplied for this transport through the protein?
So if the transporter is equilibrative, it's simply the concentration gradient that starts outside or alternatively inside and equilibrates the concentrations. If they're concentrative, they're either ATP linked or more commonly secondary ion linked. Sodium would be most common in mammalian systems, sometimes protons. Antiporters, where one goes one way and one goes the other, are much more common than people widely believe. I didn't comment on it when I showed you those pictures of all the molecules going into and out of different cell lines, especially out of. But they're not going out because the cells are leaking. They're going out because the cells are sending them out, and they're sending them out in exchange for something else, and they're doing it for reasons. And that's a whole area of biology that's been completely ignored, the idea that cells are sending out small metabolites for signals. There was just a paper in Nature not too long ago that mentioned it too. But in my opinion, antiporters are massively important. They're also important in biotechnology when you want a bug to spew something out and take something in. If you can arrange that those two are coupled, then it's good news. And by the way, they often are.
Okay, thank you.
So I think that we'll wrap up the Q&A now. I think it's been a very thought-provoking session. Certainly make you think a little bit about how your drugs get on board and how they get on board in terms of toxicity, phenotypic screen. So I think there's a lot of thoughts there, and I think the scientific community will hopefully react in the right way and continue to innovate and look at these things. So once again, thanks to Douglas for joining us, and thanks everyone who's engaged with the Q&A, and to those of you attending. I'm just going to share with you once again for those who didn't see it. So this will be just highlighting that next week we will be going back more traditionally to chemistry. So let's go back there. So we've got 2 talks from AstraZeneca, are more process-driven talks. So back-to-back talks from Matthew Burns, John Mark Henry, and then following up on the 23rd of June for a MedChem presentation from Bilayer. So please sign up for these events. We're here for you to in the community to share these presentations and engage Q&A. So I hope you can join us for those in the meantime. I will close this this session, and I hope to speak with you in the near future. Thanks very much.